Once upon a time there was a steroid hormone, which is a lipid communicator. It went through the lipid bilayer because it is a lipid. The cell membrane is composed of a hydrophobic, nonpolar lipid bilayer and is impermeable to large and charged molecules. Since the hormone and the membrane are both composed of lipids, the hormone could sneak into the cell and go through the membrane of the nucleus all the way to the DNA code after it connects to a receptor of the target cell. The hormone bound to a protein receptor and went to the nucleus. The hormone was a message sent to transcribe the code of one segment of the DNA, or what we call a gene. When the gene was turned on, it could begin the ultimate process of genetics, which was to create a protein. In the nucleus, there's DNA. DNA is composed of nucleotides which contain a sugar, a phosphate group, and a nitrogenous base. The bases of the rung are the rungs in the ladder and what we think of when we think of the letters of the code. During mitosis or meiosis, aka cell division of the nucleus, the DNA winds itself into structures called chromosomes, which are easier to pull apart and isolate into separate cells. Human DNA contains 6 billion nucleotides in order to make 25 to 35,000 genes. The code would fill a book with over 4 million pages. DNA is transcribed into RNA. This is turning on a gene. During interphase of the cell cycle, the hormone receptor complex works as a transcription factor by assisting in the folding of the uncoiled DNA strand. When activator proteins bind on the enhancers, the DNA folds on itself so that the enhancer meets up with the promoter sequence of DNA. As that happens, hundreds of different types of transcription factor proteins form a complex to make it easier for the protein enzyme RNA polymerase to attach the DNA strand and begin binding nucleotides together to create a complementary RNA molecule or mRNA strand. Only 2% of human DNA is transcribed into messenger RNA in order to turn on or express genes by making proteins. The other 98% of non-coding DNA transcribes ribosomal RNA or transfer RNA, as well as other regulatory RNA such as microRNAs or small nuclear RNAs, microRNAs which help break down messenger RNA in the cytoplasm in order to stop or turn off the expression of a gene. Small nuclear RNA is often used to splice the messenger RNA and process it prior to having it go to the ribosome. There are also large portions of non-coding DNA that scientists have not yet discovered its function. The messenger RNA continues to grow off of the DNA. RNA polymerase can build chains of messenger RNA that are up to 2.4 million nucleotides long, as in the case of the gene that codes for the protein that attaches muscle fiber cytoskeletons to cell membranes. RNA polymerase ends transcription and releases the messenger RNA when it runs into the terminator sequence encoded at the end of a gene on the DNA code. The messenger RNA is further processed by snipping out introns and splicing the exons back together to make the actual recipe for a protein. Messenger RNA leaves the nucleus. The messenger RNA leaves the nucleus in search of a ribosome. Before it leaves, it has caps and tails put onto the processed exons of the messenger RNA. Ribosomes are made of ribosomal RNA, rRNA, and proteins and are assembled by the nucleolus inside the nucleus. When the messenger RNA leaves the nucleus, it finds a ribosome. Transfer RNA brings amino acids to the ribosome to match up with the messenger RNA. At the ribosome, the mRNA nitrogenous bases are read in groups of 3 or triplet codons. Transfer RNA or tRNA contains complementary anticodons which specify for a particular amino acid. The 20 different types of amino acids are connected in unique arrangements based on the sequence of messenger RNA. The RNA is read and more amino acids are joined together. This is translation. Transfer RNA continues to bring amino acids, which continue to be assembled into what is called a polypeptide chain, as the messenger RNA strand continues to be read at the ribosome. This process is known as translation. The messenger RNA strand will continue to rebind to more ribosomes, making more copies of that protein, until microRNA degrades the mRNA strand or blocks further translation. This is where the gene is turned off. The amino acids coil up to make a protein. The primary polypeptide structure will coil and fold into its secondary structure, which then forms a functional shape as a particular amino acid in the chain interacts with other amino acids. Therefore, the particular unique sequence of amino acids determines the ultimate shape, and the shape determines the unique role that protein will perform, from enzyme to receptor to antibody to tissue fiber. If multiple polypeptide chains come together, the protein then has a quaternary structure. Free-floating ribosomes make free-floating proteins which stay within the cell. If that protein was created via a free-floating ribosome, it is destined to fulfill a role inside the cell, such as a structural protein as a part of the cytoskeleton, or as an enzyme or pump within mitochondria, or as an enzyme within a peroxisome. Proteins make up your cytoskeleton. Structural proteins provide a framework and general shape for the inside of the cell. They attach to the cell membrane as integral proteins. Actin and myosin filaments are proteins that ratchet past each other to contract the overall length of muscle fiber cells. Structural proteins also form the basis of support in connective tissue. Connective tissue such as bone, cartilage, tendons, and ligaments consist of cells, ground substance, and fibers. Collagen protein fibers make up to 90% of connective tissue, and collagen protein makes up about 30% of all the protein in your body. Proteins make up the spindle fibers and motor proteins in cell division. The spindle fiber is sent out from centrosomes to grab the sister chromatids during cell division are made of proteins. In addition, motor proteins are responsible for the movement of the chromatids during anaphase. Your body makes 3 million new cells a minute, which means that 6 billion nucleotides get replicated 3 million times a minute. Wow. Proteins act as enzymes to break things down and build things up in mitochondria and move protons across membranes in order to get the energy from the bonds in our food into usable energy in the bonds of ATP. Proteins exist as enzymes which lower the activation energy needed to do chemical reactions or form and break chemical bonds. In cellular respiration in mitochondria, enzymes help break the bonds of sugar and build the bonds in ATP. Proteins are also responsible for the active transport of hydrogen ions across the inner membrane to high concentration, as well as their facilitated diffusion through the channel of ATP synthase. The energy stored in the bonds of ATP is used by your brain and nervous system. The active transport of ions to create action potentials in electricity requires ATP. Although our brain only makes up 2% of our total mass, it uses 25% of the total energy. The sliding of protein filaments past each other to contract muscles requires ATP. This is not only important for moving our skeleton, but is required in the muscles of our heart and diaphragm. In addition, ATP plays a role in the movement of organelles and chromosomes during the 3 million cell divisions that take place every minute. There is a lot of work happening every second in our body. The energy in the bonds of ATP transferred from nutrients like glucose during cellular respiration provides the ability to do that work. Proteins also function as enzymes and pumps in plant cells for photosynthesis. Enzymes and pumps also exist in chloroplasts in plant cells. Here they assist in the breakdown of water and buildup of ATP in the light-dependent reactions and the buildup of carbohydrates in the Calvin cycle. They also function as pumps and channels in the thylakoid membrane. Proteins can also stay inside the cell as enzymes and peroxisomes to break down reactive oxygen species. Proteins can also exist as catalase or peroxidase inside peroxisomes to break down hydrogen peroxide into water and oxygen. Hydrogen peroxide and other reactive oxygen species are a byproduct of metabolic reactions and are also produced by leukocytes as a cytotoxic chemical to help destroy pathogens. The body needs to clear these extra free radicals so that they don't harm our own cells by stealing hydrogens from the fatty acid chains of membranes, thus creating a chain reaction of oxidation and ultimately oxidative stress, which can lead to heart disease, cancer, or diabetes, amongst many other problems. Starting over back at transcription, messenger RNA can also go to ribosomes on the endoplasmic reticulum. These proteins are destined for functions outside the cell. To start back over at transcription, messenger RNA does not always go to ribosomes that are free-floating to make proteins that stay inside the cell. The messenger RNA can also go to ribosomes on endoplasmic reticulum to be packaged and processed in order to work outside the cell or with substances from outside. At the rough ER, proteins are translated and packaged into vesicles. At the rough endoplasmic reticulum, ribosomes on the rough endoplasmic reticulum, proteins are translated with messenger RNA, RNA, rRNA, and tRNA. The amino acids are linked into polypeptide chains and packaged into vesicles. The proteins can be modified within the rough ER vesicles. The Golgi apparatus modifies the proteins and directs them to their targets. The vesicles from the endoplasmic reticulum drift through each layer of cisternae of the Golgi apparatus. Here the proteins are modified and have molecular identification tags added, such as phosphates or carbohydrates, so that they are now targeted for unique destinations and interaction with other cells and molecules. One such role of proteins in this situation can be antibodies which fight disease. One such classification of protein is an antibody. Antibodies attach to specific antigens on the surface of foreign pathogens and agglutinate them together so that eosinophils and other leukocytes have an easier time destroying pathogens before they can cause an infection. Proteins can also be hormones to communicate, receptors to receive communication, or anchors which hold cells together. Proteins can be non-steroid hormones which are sent out to communicate with other cells. Proteins can also be receptors that receive hormones to activate enzymes, open channels, or transport messages to the nucleus. Proteins can also be embedded in the membrane to link together cells and epithelial tissue. Some hormones that communicate can be neurotransmitters or ligands. Proteins can be embedded in the cell membrane as channels or pumps to help with facilitated diffusion or active transport. Proteins can also be embedded in the cell membrane to assist in the transport of molecules across membranes. They can be channels that facilitate diffusion and allow large or charged molecules to move from high concentration to low concentration. They can also be pumps that use energy to actively transport molecules against the concentration gradient from low to high concentration. Proteins can also be enzymes and lysosomes. Proteins that are enzymes and lysosomes help lower the activation energy for the hydrolysis of nutrients. The enzymes and lysosomes can also function to break down old or damaged cellular organelles. Proteins get turned off by other proteins. Enzymes tag proteins to be broken down. Once tagged, a proteasome, which is a protein, engulfs, uncoils, and breaks the peptide bonds of the protein. The process of protein synthesis is coordinated by many organelles within the cell. Ultimately, proteins run the show. Proteins are the most abundant and diverse organic compounds of the human body. There are roughly 100,000 different kinds of proteins. and they account for about 20% of the total body weight. All cellular structures and functions are determined by proteins. Structural proteins determine the general shape and internal structure of a cell, and enzymes direct the cell's metabolic activities. Hormones alter cellular operations by changing the identities, activities, or quantities of important enzymes and structural proteins in various target cells. The sensitivity of a target cell is determined by the presence or absence of a specific protein receptor with which the hormone interacts. Life as we know it would not be possible without proteins.